So thank you very much, Tony, for those kind words, and many thanks to the organisers for inviting me. It's a pleasure to be here. I haven't been to this part before, so far I'm enjoying it very much, although it's a very brief visit. So this is the... Is this which... Do I need to do anything with it? Okay. Right. Okay, so this is the title of my talk. It may seem a bit arrogant to say that modeling transmission is the key to understanding. I think transmission is a key to understanding because however you look at a virus epidemic, disease epidemic, somewhere in there you've got to get the elements relating to transmission right. And if you can do that, you can show all the other aspects that interact with it. Just to comment on a few of the names, Glenn Powell is an entomologist that used to be at Imperial College. Nick Cunniff is a biomathematician at Cambridge. John Holt is mainly involved in quantitative risk assessment nowadays. He's also involved at Imperial College. Larry Madden at Ohio State University. Neil McRoberts in California. And most of all, my longtime colleague and friend, Frank Vanden Bosch. We had a lot of discussion yesterday about the different transmission types, so I thought it'd be useful to show what you can see is quite an old slide. And this really sets out the 4 different transmission types that you can have. So non-persistently transmitted, if you want an anatomical explanation of that, it's basically stylet borne, but that does not mean it's just a contaminant. There is often a very precise molecular interaction with a receptor on the stylet and the virus within plant cells. Indeed, some evidence suggests that when, for instance, an aphid is probing, the presence of the stylet is sensed, put it well, you know what I mean, is sensed by the virus, and the virus changes its conformation so that it's more likely to be to meet with the receptor on the stylet. Semi-persistent transmission, the virus actually moves to the foregut of the insect. It doesn't go any further, and it may get regurgitated from the foregut. And then we come to the persistent transmission classes, the first of which is circulative, which means that when the virus is acquired, it's able to go from the stylet to the various guts within the insect, and then to pass to the hemolymph and then recirculated back to the feeding parts, so that it can be then re-inoculate a plant. The difference with the propagative persistently transmitted class is that the virus can actually replicate within the insect. So we'll come back to some of those aspects later in this talk. But the importance of these transmission types is basically these types govern the rate process that are involved. So the acquisition rate, the rate at which a virus is acquired, the latent period, this is really the time delay from the time of acquisition to the point where inoculation of another plant can occur. With a non-persistent virus, that can be very short. With the persistent classes, it can be days. It can be it can be several days. Then there's the rate of inoculation, which may be affected in some ways. There's the period of infectiousness that can be affected by the transmission type. If a virus is acquired in a non-persistent way, generally it can then participate in maybe 2 more inoculations. So it's a very limited period of infectiousness. Whereas with the persistent transmission types, effectively, for many insects, it can be the lifetime of that insect, although the actual virus within the insect may decline over much of that period. And then again, for the persistent transmission types, we have the phenomena of First, transavarial, I'm calling it here passaging because it's probably a better term to use, whereby the virus can go to the progeny, can passage to the progeny. And there are other cases where if it's acquired as a larva, the virus, then it can passage through the different stages that the insect goes through until it becomes an adult. So these last two features are important in the persistent type of transmission. So, approaches to model plant virus epidemics. I'm not going to say anything about, there's been several attempts to use complicated simulation models to do this. I'm not going to cover any of that material at all. Basically, the approach that's been taken is to use SEIR type models. In some cases, well, in a lot of cases now, to link those essentially plant-based SEIR models with vector models which fall into a similar sort of categorization. And then the question is, when do we make it more complicated? When do we simplify? And that very much depends upon the problem definition and the way you formulate the particular modeling approach. We also heard some talk yesterday about symptoms and infectiousness. This is just really to show how you can set the structure for an SER type model. Basically, here we have two lines here, two thresholds. There's one which is a threshold in terms of virus titer for when a vector can acquire a virus from a plant. The other dotted line here refers to, it's a symptom visibility threshold, and that's in relation to symptom expression. Remembering that in some ways, symptoms, the assessment of symptoms is very arbitrary. You may rely on visual symptoms, or you may use other molecular-based methods to, as your basis for saying whether something is symptomatic or not. But then you have these various aspects here. So if we stick with virus titer again, there is a period where it's very low, and then it starts to increase. It then reaches a point where it's at a sufficient level that a vector that's probing or feeding can acquire the virus. The virus titer may continue to increase because of replication, multiplication within plant cells. And this period of, but then it may actually start to decline again for various reasons until it crosses that threshold again. That period of time you can call the infectious period within the plant. If the virus titer falls below that acquisition threshold, because for one reason or another, because it's exhausted the cell's resources, or because the plant is basically dead, or because the plant is changed in morphologically such that the vector is no longer attracted to it, then effectively it's removed from the epidemic development, and we enter this post-infectious period. So I think this is one way of showing how you can develop an SEIR type model quite naturally from the basic biology that you're seeing. And what you generally end up with from an analysis of these SEIR type models is a derivation of a basic reproductive number, defined here as the number of new infections arising from the introduction of one infected unit, and we can come back to that, into an otherwise healthy population during the unit's period of infectiousness. And if that value is greater than 1, then an epidemic will develop. There are problems with this. In my view, it's not always appropriate to use this as strictly defined, estimated using things like the next generation matrix, where you have multiple pathways of transmission. And that's something else I will come back to. In my view, often the most useful metric to use is an invasion threshold that you can develop, which might not be the same as the basic reproductive number, but there is often a relationship. And in many cases, some of the arguments in relation to how you can develop different forms of the R0 metric, get rather arcane, and I've discussed this with several of you already. But basically, what we're trying to do is to put together information on the virus, the vector, the plant response to that, and try and turn that into an epidemic curve. This is a nice slide from Larry Madden. And if you draw a schematic of how you can model this, on the left-hand side here we have the plant. This is a bit old, this terminology, AHL-SR. It was done at a time when I thought that really plant pathologists wouldn't understand SEIR, so I tried to use symbols which were more readily understood perhaps by plant pathologists, but I've moved right away from that now, but that's how it was done at the time. And then on the other side you have a similar scheme for the vector, where you have a healthy vector, a non-viriliferous vector. There's a period of latency as defined by the way that the virus is interacting with the vector, and you have a period of infectiousness. And then the linkage, of course, is that non-viriliferous vectors can acquire virus from infectious plants, and viriliferous vectors can infect healthy plants. So that's the important role that transmission plays. And we worked on this. We developed this original framework back then, published then, had various elaborations. This particularly looked at the way in which you can involve the movement of vectors in and out of a system, and how that affected the types of characteristics there. And there was another one where we were looking at a system where, in addition to the vector transmission, there was also transmission between taking cuttings, growing them on, and replanting them. So in this particular system, there were two pathways of transmission, which made the derivation of an overall R0 quite difficult to interpret. And nowadays, I would simply interpret this as an invasion threshold. And then in terms of management, the implications of some of these more theoretical work for host resistance or disease control more generally, there were these two papers published. Gordon, who's here, was actually involved in this particular work here when he was at Rothamsted. And the basic question was, to what extent do plant breeding methods and or improved cultivation methods select for more. I know there was some discussion about virulence yesterday. Let's keep this in a rather broad sense, the aggressiveness of a particular virus strain or species. And to do that, we looked at the various ways in which resistance could work, the availability of healthy or disease planting material, and what circumstances we might have to use disease planting material, and the time scale over which we'd be considering possible changes. So in terms of resistance, there are many types of resistance mechanisms. Some act directly against the vector. In other words, they're direct vector resistance. Some act in terms of the vector virus acquisition. So there may be no resistance to the vector, but there's actually some form of resistance to acquisition of the virus from a plant by the vector, or inoculation of the virus to a plant by the vector. And then, of course, you have the intrinsic characteristics of the virus itself, such as its ability to multiply within host cells. So there are different types of resistance that can be used, and of course there are many different methods for managing vectors, such as netting, in this case beans, to keep whiteflies from affecting these bean seedlings. So we developed firstly a population model for the virus vector, which is really no different from the previous slide I showed. It's a bit simplified, and perhaps it's done in sort of nicer colors and patterns. So these are the viriliferous vectors there. And as well as doing that, we also developed a within plant virus dynamics model, which effectively was looking at the number of virus particles produced per infected cell per unit time. So we had a system for doing that. But the question was, if you deployed different forms of resistance, different mechanisms, what effect would that have on some of these other parameters? So we asked the question, well, if you deploy a pest-based resistance or a virus-based resistance, would the virus actually be able to evolve in such a way in response? And to do that, we used generally the theory of evolutionary stable strategy. So we constructed the models together, and we considered the situation that if you have a resident virus, maybe the wild type, and then you introduce a new form of resistance at some point in time, what would be the effect, in this case, on the virus multiplication rate? And we were actually able to show by direct calculation in this case, because the model was amenable to that, that that the virus multiplication rate would increase to a new higher level in response to resistance being deployed against it. That's shown schematically. There's no timescale or anything to do with that. But that paper has been published. Then we looked at, again, these horizontal and vertical transmission routes. So, whoops. So here we have the typical vector transmission, but here also we have the possibility that this is a cassava plant. So the cassava plant gets infected, it's harvested, the canes are cut into sections, and you use those to propagate the material. And in some cases, you get a phenomena that as you take these stem pieces, in the previous season, there might have been leaves attached to them which are showing symptoms. Yet you get this property of remission, that the stems, actually, when you plant them out, they do not show virus. So there's this sort of slight remission phenomena that occurs. But nevertheless, it's another route for the virus. So again, we can go through the same process in terms of the population model, and we add in this. You can add virus-free material. You can be taking cuttings, preferably from healthy plants, but sometimes farmers may have to take them from diseased plants. And you link that with the vector transmission, and you go through the whole process again of seeing what sort of selection is placed on what selection is placed on the virus by some of these disease management options. And we looked at using virus-free planting material, vector management, such as the netting example I showed, using very stringent cutting selection to be sure what you're actually planting, and roguing, or as I've sometimes misspelt it, rouging. And I won't go through the outcomes in detail, but it turns out that vector management, in the sense of doing things like netting and other aspects, does not put a selection pressure on the virus to increase its multiplication rate. Roguing actually selects for low virus titer. And you can interpret that in terms of, well, if there's a low virus titer, then the virus is not going to get rogue, because often virus titer is associated with a level of symptom expression. However, some of the counterintuitive results that we got were both cutting selection and in vitro propagated material, which are the prime features that are used in most propagation schemes for plants which are propagated in this way, actually does put a large selection pressure on the virus to increase its multiplication rate within plants, given time. Roguing is simply inspection and removal of diseased plants. We decided that we wanted to do a little bit more work on within plant virus dynamics. We wanted to see how simple we could do this, so we could link these two aspects. So, typically we would have the number of plants in a population, we'd have a number of infected plants. Then we'd have this measure of the total virus load within a particular plant population. And if we could then specify what the maximum virus titer per infected plant could be, then we would have a parameter there, which we could use to actually derive some surrogate variables. The first of which is a variable which is the mean virus titer per infected plant, which we can call C. And we could construct this variable, which basically is V, the total virus population, divided by the maximum virus titer per infected plant. What that gives us is the equivalent number of plants that would be at maximum virus titer. Obviously a value which is much less than just I, for example. And you can express both of those in relation to the original variables. And then you can construct a model in which, in addition, you include virus limitation. In other words, to what extent has the virus multiplication in your average plant reached its maximum and can't multiply anymore, just as you can include host limitation, where the number of infected plants is approaching its maximum value. And I won't go into the maths. Again, it's been published. But what is nice about it, seemingly, is that you can actually get a closed form solution for C. Well, not for C, for T in terms of the concentration. So from this, you can predict the length of time it would take for a plant to reach its maximum virus titer. Sorry, that is scaled virus so 1 would be the maximum virus concentration in a plant, and that would take you from the initial concentration to the maximum, and this would be able to tell the time it would take. So in this case, it pretty much goes to saturation. However, what we find is that if we plot these various things, so this is infected plants, virus this is the virus, overall virus load, and this is the mean virus concentration per plant. This gives a typical plot for it. This is a composite parameter which is mainly determined by the actual contact transmission rate in the model. And as you slightly increase that model, that value, you find that rather than declining, as in the first instance, it reaches a plateau just for a small change in the contact, and then it goes into a rapid increase, where the concentration is basically staying the same. So you ask yourself how valuable that sort of information might be. But we ask the question, well, if you can collect field data on numbers of diseased plants and the mean virus titer in those plants, can you then make estimates of what the between-plant transmission would have been and what the within-plant multiplication rates are? And the reason we wanted to do this, because often in the literature, well, it's not often, it's often in the grey literature or reports of one form or another, you get reports of very few occasional diseased plants, heavily diseased. Vectors are active, they're present and active, and yet you get these isolated single plants or a few plants by themselves with no further expansion. So from this work, we were able to develop whether it's actually virus limitation which might determine an invasion threshold. Because there are two predictions from this. Is if you don't meet the threshold, then what you would end up with would be a relatively small number of diseased plants at a high virus titer. On the other hand, if you do meet the threshold, then you would have many diseased plants, but each of those, the average virus titer in this large number of diseased plants would be relatively low. So it's a contrast which may be able to explain this phenomena of having small number of individual plants at high virus titer, vectors are active, but you don't get any spread. And I won't say anything about the last point. Some other aspects relating to transmission. We don't actually capture all the information. For example, this whitefly here, which acquires and transmits Bergamo virus in a circulative manner, it's been claimed that you can get horizontal sexual transmission between vectors. Now, this is a phenomena which is very well known in insect viruses. It's a bit contentious, it's a bit controversial whether this can be repeated, but there is this claim that you can get horizontal sexual transmission. We certainly know that we can get transovarial transmission, although in whiteflies, again, this is a bit controversial because this particular group said, yes, you can get it, but the actual virus ends up as being not being effective, not being efficient. It's lost some of its properties. And the other aspect, really, this broader aspect, is what are the effects of natural enemies of vectors on transmission? And I think it's useful just to step back and take a different view of plant viruses. Now, this is a view which really comes from an anthropocentric view. It's taken the view of the vector. So, for example, we can have horizontal transmission, which with the vector can be contact or venereal. We can have vertical transmission, which can be transovarial, involving the various ways in which insects can reproduce. And then, of course, we can get indirect transmission. Only in this case, and this is obviously the case for the persistent class of transmission, we're having horizontal transmission from insect to insect using the plant as a vector. So this is simply a different way of looking at some of these plant viruses, and I'll come back to that right at the end. When we modeled this, this system where we had sexual transmission and transovarial transmission, we ended up with what we then called a basic reproductive number R0. Some would say it's an R0 squared. I don't think it satisfies either condition, because here we have the indirect transmission, which corresponds to plant to vector and then from vector to plant, which would be the R0 squared element of it. But these two are transmission events within vectors, so they are not R0 squared terms. So these are the direct transmission terms through sexual transmission, and these are the transmission terms through transovarial transmission. And we did some work on this where we computed the elasticities to look at the various importance of some of these different transmission types. Sorry, I should have shown that. We have two, of course, because you can get transovarial transmission both through the sperm and through the egg. Natural enemies. Again, this is published work, and we start off with the basic premise that biocontrol has shown that parasitoid predators can decrease vector populations. There's also some experimental evidence that actually introducing this higher trophic level does not necessarily mean a decrease in viral disease transmission. And the evidence, there is also a lot of evidence of some of these complex signaling devices where, for example, you can have an alarm response where if a vector is on a plant, the plant responds by sending out a signal which will attract a parasitoid. We have a vector-host plant preference signaling, which may determine whether a vector lands on a healthy or infected plant, maybe because of some volatile signaling. And then we also have this cry for help response. Sorry, I've got this the other way around. This is the cry for help is the plant crying out for the parasitoid to come along and protect it from these nasty parasitoids and predators. This is the alarm response here is the signaling between the vectors to say a parasite has landed, so get away quick before it attacks you. So all of these signalings can affect transmission. And what we did, we come up with a set of equations. There were 7 equations, one for the host, 2 for the host, whether healthy or infected. And then we have 4 categories of vector, depending on whether it's viriliferous and carrying a parasitoid or not. And then, of course, we have an equation for the parasitoid population. And we ended up with a rather complex, again, I might be criticized for saying this is an R0. Okay, it's an invasion threshold. But the important thing is, when you look at these terms in succession, you can interpret each of them. This term then relates to a paratized vector that acquires the virus. This term relates to an unparasitized vector that acquires the virus, and this term, the final term, reflects an unparasitized vector that acquires the virus and subsequently is parasitized. So I think that's what's so important in looking at these thresholds and being able to interpret them in a way that is biologically intelligible. And you can do some more work with these again, R0s. So this just represents two cases. So on the left here, we have a system where the R0, without the effect of the parasitoid, there's a high value of the R0 value. So it's a pretty successful system there. This is the virus extinction threshold at 1. So when you then introduce the parasitoid, what you find is that at very low levels of the parasitoid, actually the R0 is substantially increased, but eventually there comes a point of the parasitoid density where you're below the virus extinction threshold. But perhaps the more interesting case is this. If you don't have the parasitoid, then the R0 is less than the virus extinction threshold, so there's no disease. But then you introduce the parasitoid and you can rapidly exceed it. So actually in this case, introducing a parasitoid has turned a situation where there is no epidemic developing into one where it does, depending on the parasitoid density. Now, I've been criticized by the biocontrol community for this, for saying that biocontrol doesn't work. I'm saying nothing of the sort. It does work eventually. All I'm saying is that under some circumstances, because it's probably increasing the mobility of the vectors, it's leading to enhanced transmission, and up to a certain point, it's actually increasing the virus spread. Just a bit of mention of some current work. Tospoviruses are members of the Bunyaviridae. It's the virus family. Many members are either insect viruses or have origins of insect viruses. And in Tospoviruses, these are transmitted by thrips. And it's a rather peculiar case because only juveniles acquire. So juveniles only acquire virus if they land on an infected plant. So the actual status of a larva, of a thrips, depends on the plant it was born upon. Okay? And then the virus can pass transstadally through the various molts of the larva to the adult, and the adults can inoculate. Adults do not acquire. During this period of development from the larva to the adult, there are anatomical barriers, which means that the virus cannot actually act in the way that it can get back to the mouth parts again. Adults can acquire, but they cannot complete the cycle. So the adults can pick up the virus, but it never gets back to the feeding part so that it can then inoculate. And we got to thinking about some of the evolutionary implications of this. So we developed a model for the Tospovirus case. It was a system of 6 equations. We had the plant population, healthy or infected. We had equations for the larval population. We simplified this simply to a juvenile population, viriliferous or non-viriliferous, and an adult, viriliferous or non-viriliferous. So we went through the standard procedure for developing an invasion threshold by constructing the invasion matrix. In the Tospovirus case, it was a 3x3, and developing the invasion threshold. We did the same for the case, which you can call non-Tospovirus, where there is only adults involved in the transmission process. And that actually led to a 2x2 invasion matrix. And we also looked at the case in which, well, let's suppose the Tospovirus, by whatever means, manages to acquire the phenomenon of adult acquisition. What would the outcome then be in terms of an invasion threshold? And finally, we tried to identify maybe some fitness costs which would make it difficult for the thrips Tospovirus case to acquire the characteristics of adult acquisition. So we looked at that. I'm putting up this in full because, again, it shows to me how important it is when you derive a threshold, or an R0 for that matter, that you can actually go through the sequence in words to explain what's actually going on. So in this case, for the Tospovirus, we start off with an initial population density of the larva per plant, the various acquisition probabilities, becoming an adult, the plants visited per adult thrips per day, inoculation probability of an adult, average lifespan of adult vector in relation to the number of healthy plants and the average lifespan of a plant. So heuristically, I'm convinced that this invasion threshold, whether you call it an R0 or not, I'm not bothered. It does tell us something really biologically useful. In terms of these trade-offs, this is a simple convex relationship between a trade-off E1. Let's start with E2. Let's suppose that the Tospovirus case is starting to acquire the ability to acquire as an adult. So even a small amount of gaining of that ability will lead to a major loss in terms of its ability to acquire as a juvenile. So there's a trade-off there which you can show has to be a convex relationship, because what we're trying to do is, let's assume that for the Tospovirus case and the not Tospovirus case, that in fact both of those invasion thresholds are greater than 1. What I didn't mention is when you combine all the characteristics, all you get is the sum of the two component invasion thresholds. So the interesting thing is, well, if that's the case, why doesn't the system acquire the ability to inoculate to acquire as an adult? Because that would give you a higher invasion. It would make it easier to meet the invasion threshold. So that's why we started to look at these as particular trade-offs. And indeed, you can show that. So, for example, on the left, this is the invasion criteria for the Tospovirus case. This is the invasion criteria for the non-Tospovirus case. And these are just 3 selected examples over the fitness cost. And remember, there is a trade-off involved, so I only need to plot it against E1, which is loss of the ability as to acquire as a juvenile. There is a region here where, in fact, there is a real constraint on acquiring this additional characteristic. Well, that's just 3 particular examples. The zetas are the invasion thresholds. You can actually do a little bit better. You can come up with a type of contour map, which you can do in various ways. So this is the Tospovirus invasion threshold. This is the non-Tospovirus invasion threshold, and these are the fitness costs you have to use such that the overall invasion criteria threshold for where you have the combined characteristics is less than 1, where the 2 component ones are greater than 1. And this undefined space really is just where you have complex solutions to the relation defined for the trade-offs. Just a couple of words on vector preference. That's come up a couple of times at this conference. Does it prefer to visit an infected or a healthy plant? This is just a backdrop. I'm not going into this, except to point out that you need to distinguish between the landing preference and actually the settling preference. That's a key thing in this, because the argument is that, you know, if it lands and it probes, but then is repelled and moves on until it finds something it can settle on and actually stay and feed on, that's what the actual process which might have an effect on the transmission and the epidemic development. But there are some key questions, I think, in terms of vector preference. What effects does it have on epidemics? Well, there's been previous work done by Sonny and by Mark Sisterson and by some recent work in California on this. But there's still some questions that need asking. How does it vary between persistent transmission and non-persistent transmission? How does it relate to vector life history and abundance, dispersal ability, crops or natural hosts? And perhaps an interesting feature, is it possible that a vector, once it's infected, can switch in its preference? So can it switch for a preference to diseased plants, which is often the claim, to then a preference to healthy plants? Because obviously it will be to the virus's advantage to do that. And if it is the case, how would you incorporate a switching mechanism in a model? Would you go for a population-level model or an individual-based model of one form or another? I don't know. But some more interesting questions. How did that evolve? Why does it not always happen? And it's certainly the case that it doesn't always happen. And can knowledge of this interaction be used to disrupt transmission for management purposes? Okay, so just to end quickly up with some of the final challenges. Sorry I've gone over time. We really haven't got all the devices we need to deal with multiple pathways of transmission, if that's what we need to do. If they're present, then we need to do it. I'm not convinced that there's a lot of work to do in developing the concepts of R0, the R0 squared, the type R0, the R0 which is an operation when you have some sort of control in place. My preference now, I've moved from really trying to explore R0s to actually use overall invasion thresholds and justify them in a biological way. But the challenge, I think, is also for looking at evolutionary trajectories. Most virologists consider evolution to be looking at DNA sequences, looking at base substitutions, and that's evolution. It's clearly doesn't explain what actually happens when we get disease epidemics developing. In many cases where we're looking at evolutionary trajectories, we use trade-offs of one form or another, just as I showed in the previous example. The problem is, you really have to show a mechanism for what the basis of that trade-off is, rather than just being some sort of abstract construct. Then the final challenge that I would put to you is, well, actually, in the first place, how did vector associations develop? You can imagine a situation where primeval viruses would have developed by one way or another within plants, if plants had evolved at that time, who knows? Within insects. At what point in time was it necessary to, or did it assist to actually involve a vector to assist? So, for example, a plant virus, you could imagine an ancestral plant virus would have had seed transmission, would have pollen transmission, and so forth, and be able to sustain itself in the population. But at some point, a vector association develops, so maybe it doesn't need some of these other attributes like seed transmission and pollen transmission. Similarly, you can get an insect virus. It gets along perfectly well through contact, through sexual contact, through transovarial transmission, all these various forms of vertical transmission. But at some point in time, a vector gets involved, a vector in this case being a plant, which enables the virus to get back from one insect to another insect. So how did these associations develop? And is there a role that the modeling can play in this? And I think that's a question that, to me, is a real challenge. Thank you very much.